We're going to take a look at the blood vessels, especially the arteries and arterioles. So remember that the arteries conduct blood away from the heart. Capillaries are where we're going to be doing the exchange between the blood and the tissue. And veins are going to be conducting blood back towards the heart. So up at the top we show the blood flow through the systemic circuit, going from the left ventricle out through the arteries, and then the arteries will get smaller and turn into arterioles, and then the arterioles will feed into the capillaries where we do the exchange, and then the capillaries drain into the venules, which then open up into the larger veins, and then the deoxygenated blood will be taken to the right side of the heart to be pumped to the lungs next. Here's a picture of the three types of blood vessels. We have arteries on the left, and they're going to feed into the arterioles and then the capillaries, and then the capillaries will drain into the venules and then finally into the veins and back to the heart. So if you take a look at the difference between the arteries and the veins, you'll notice two things that really stand out. So one is the thickness of the walls. So notice that the arterial walls are very thick and the walls of the veins are much thinner. And that's because the arteries are close to the ventricles where the high pressure is being generated, and so they've got to be able to withstand that high pressure. So artery walls are going to be a lot thicker. And remember, the pressure inside the veins is much less, so their walls don't need to be as thick. The other thing that's different with the veins is that they have valves in them. So the valves are going to prevent the blood from flowing backwards. Because the pressure is so low, it's very easy for the blood to flow backwards in the veins. So the valves will stop that. And then this animation over here, it's kind of hard to see, but you can actually see the valves inside this vein opening and closing. And then the capillaries, where we're going to be doing the exchange with the tissue, they're very thin-walled. They only have one cell layer thick. So it's very easy to do exchange with the tissues at the capillaries. This microscope view shows that the walls of the artery are going to be much thicker than the walls of the veins. The other thing that's nice about these pictures is that you can really see the layer of smooth muscle that surrounds the lining of the arteries and also in the veins. So the smooth muscle will contract and relax, and that'll control vasoconstriction and vasodilation. So let's take a closer look at the arteries. They do have a larger diameter than the arterioles. So what does that do to their resistance? So if you think about the wide straw versus the narrow straw, so arteries are going to be the wide straw, so they would have low resistance. And if they have low resistance, then that means that they have high blood flow through them. So it's very easy for blood to flow through the arteries. And we talked about how the arterial walls are very thick, and that's because they have to be able to withstand the high pressure coming from the ventricles, which they're close by to. The arteries also have low compliance, and compliance is just a measure of how easy it is to stretch something. And if you have low compliance, that means it's hard to stretch. So it's like one of those hard to blow up balloons. That's an artery. So they can withstand that high pressure. They're not going to balloon out. They're hard to stretch, but they can stretch a little bit. They are elastic and they will recoil. And so they're going to be serving as what are called pressure reservoirs. So let's just take a look at how the arteries are pressure reservoirs. The top picture is showing the heart in systole. So the left ventricle is contracting and ejecting blood out through the aortic semilunar valve into the aorta. So we've got a lot of blood coming out into the aorta, it's high pressure, and so it's going to push on the aorta walls and stretch them a little bit. But remember, the aorta walls have low compliance, so they're not going to stretch a whole lot, but they do stretch a little bit. And then during diastole, when the left ventricle is relaxed, It's no longer squirting blood into the aorta, and the aortic semilunar valves are closed. And so the walls of the aorta are very elastic, and so because they were stretched a little bit during systole, now they're going to recoil and push on that blood that's in the aorta, and that will keep the pressure up in the aorta and keep the blood flowing through the systemic circuit. So that's why the arteries are called pressure reservoirs. So I would star that and make sure you remember that arteries are known as pressure reservoirs. Arterioles are going to branch off the arteries, and they have smaller diameter than the arteries. So what happens to the resistance? So remember that the smaller the diameter, the higher the resistance. So arterioles are going to have a higher resistance to flow than the arteries did. So the arterioles have the greatest resistance to blood flow, so I would star this. This is really important to remember. And we can actually regulate that resistance in the arterioles by either contracting or relaxing the smooth muscle and causing vasoconstriction or vasodilation. So we're going to regulate the resistance in the arterioles by either vasodilating or vasoconstricting the arterioles, and that's going to control two things. So one is it's going to control the blood flow to the capillary bed in that organ. So for example, if you vasoconstrict the arteriole going to an organ, that would increase the resistance, and then that would decrease the blood flow to that organ. So maybe that organ is not as active. But maybe the organ is more active now, and we need to increase the blood flow. So if we vasodilate and decrease the resistance of that arteriole, then we would increase the blood flow to that organ. And then the other thing that we can use the resistance in the arterioles for, vasodilating and vasoconstricting, is that this will help us regulate MAP, or blood pressure. So to solve for MAP here, we've just rearranged our flow rule equation and just solved for MAP. And so you can see that if you increase the resistance by vasoconstricting, that will increase the blood pressure. And if you vasodilate and decrease the resistance, that will decrease the blood pressure. So the way I think about this is if you have a garden hose, and if you squeeze the hose, increase the resistance, you've decreased the diameter, that would actually increase the water pressure coming out the hose. And then if you stretch the hose out, increase the diameter, decrease the resistance, that would decrease the water pressure coming out of the end of the hose. So that's just another way to think about this. Okay, star this graph. This is really important to understand. It's just showing you the blood pressure in the arteries. So for example, like the aorta here. And notice that the pressure in the arteries really fluctuates, and that's because it's very close to the ventricle. And so you can see that systolic, the highest pressure, and then the diastolic pressure, the lowest pressure in the arteries. And then we go from the arteries, we're going to branch into smaller arterioles. And then from there, we'll go to the capillaries, do the exchange with the tissue, and then that will drain into the venules and then the veins. And so you can see that the pressure gradually drops from the arteries, which are close to the ventricle, which is generating all the pressure, until you get to the veins. And the pressure drops because of the resistance, because you've got to travel this long distance and there's a lot of friction along the way, and that will decrease the blood pressure as you go. The other thing to star and point out here is that resistance is greatest in the arterioles. And because of that, we get the greatest drop in blood pressure out of all of the different types of blood vessels. So arterioles have the greatest resistance to blood flow, and they're also going to have the greatest drop in blood pressure here. Okay, as far as regulating the resistance of the arterioles, we're going to be either relaxing or contracting the smooth muscle, so either vasodilating or vasoconstricting. And that's going to be controlled by two things. The intrinsic factors are just things that are in the local area of the blood vessels. So these would be local factors, for example, like oxygen and carbon dioxide levels. And that's going to be responsible for regulating blood flow to that organ or that area. So for example, if an organ starts working harder, using up the oxygen, generating lots of carbon dioxide, that will cause vasodilation because we need to increase the blood flow to the area. And remember, that goes back to the flow rule again. So if you vasodilate, decrease the resistance, that'll increase blood flow to that area or that organ. We also have some extrinsic factors, things that are outside the blood vessels that will help regulate the MAP or the blood pressure. So we do have sympathetic neurons that innervate the smooth muscle of the blood vessels, and for most of the blood vessels it's only innervated by sympathetic neurons. There's not too many blood vessels that are also innervated by parasympathetic neurons. And then we do have a few hormones that will affect smooth muscle contraction and relaxation as well. So one of those being epinephrine. We also have ADH and another hormone called angiotensin II, which we'll talk about later. Okay, and as far as controlling blood flow to the organs, it's going to be based on need. So if the organ is working harder, we need to increase blood flow, so we would vasodilate to it. And if it's not working as hard, we would vasoconstrict because it doesn't need as much blood. And so to answer these questions here, how do you vary the resistance of the arterioles? And remember, that's just by vasodilating or vasoconstricting the arterioles. And this is in response to local factors, so things like oxygen levels, carbon dioxide levels, that'll affect the smooth muscle and cause it to either constrict or to dilate. We can also alter the organ's blood flow, not only by changing the resistance, vasoconstricting or vasodilating to that organ, but also what if you change the blood pressure? So what happens if your blood pressure is too low? Notice that if you decrease MAP, you decrease the organ blood flow. And then if you increase the blood pressure, then you actually will increase the blood flow to the organ. And this is just an example just showing you that you can vary the resistance to the organ by vasodilating and vasoconstricting, and that'll affect the blood flow. So for example, if we look at organ B here, here is the diameter of the arteriole feeding into organ B, and here's our blood flow of 1 liter per minute. But if we vasoconstrict to that organ, maybe now it's not as active. If we vasoconstrict, we increase the resistance, and that'll decrease the blood flow down to 0.5 liters per minute.